Uh, oh, yeah. Not even like in the garage or outside. Yeah. Me, so I'm in North Carolina in the U.S. So raccoons, yes, we have raccoons, we have deer, geese, all throughout a little subdivision. It wasn't clear cut when I where I am right now, so there's a lot of little pockets of forest. So lots of interesting creatures go by. The source for most of my samples for my microscope is the dirty pond that's just down the street. So yeah, oh, it's amazing. Absolutely. Um, yeah. Yeah, so I'm a postdoc, so I have my PhD. Now I've moved on. James, I think you're technically done with your postdoc, right? Or are you in your transition period before professorship? Yeah, yeah. But, oh, that's so awesome. Yeah. Hmm. What what percentage of the equipment that was left there do you think is from the 1980s? Like 50, 70%? Ah. Hmm. Oh, well, you can get military surplus. Yeah. I recently, I recently, and this is actually sort of related to the book just a tiny bit. I recently went to a conference that was a reunion, more or less, from my old department at my old university. And someone came and he was supposed to give a talk, but really what he gave a talk on was the shenanigans they got into in lab. And he talked about how his boss in the 1980s loved to buy military surplus equipment for the lab, leading to one time where he set up a DNA sequencing gel, which back then was a huge apparatus that used radioactivity to follow the DNA. He cranked up this old military surplus voltage meter to 2,000 volts, way more than necessary to kill you. Then he left to go protest Reagan. He comes back about 3-4 hours later to find that the little arrow thingy had gotten stuck and actually was something more like 4 to 8,000 volts. The entire gel apparatus had boiled and the glass had exploded, spraying radioactivity throughout the lab. Not great, but it was the 80s, so, wasn't the end of the lab. They just mopped it up and called it good. Well, technically he was a professional, so yeah, not really sure. Eh, yeah, yeah, more or less. Um, yeah, yeah. They tell you how you feel about your own disease. Yeah. So, I turned you up. I got a little bit of a mention that you both of you were a little quiet, and no one's mentioned anything since I changed it. So I think you're good. I'm awful with the audio side of this, so the little level doesn't go quite into the red. So I think you're good. But what that means? Ah. I'm getting everyone sounds great from the chat, so that's good. Yeah, so I'll monitor the chat if anyone has any specific questions or comments. I'll say things out. Yeah. I never finished introducing myself though. So yes, postdoc. So I do research. I study the genome specifically in Drosophila, so fruit flies. But we're close enough to Drosophila, plenty closer than any prokaryote, definitely. Um, but yeah, we study the genome, so we do all sorts of things. Dropping thousands of dollars on experiments every other day. I also teach partially, so my postdoc is part teaching. So I teach at an underserved university about 2 hours away during the semester. I can, I just don't want to. Yeah, yeah, no. They serve a historically underrepresented community, and that's sort of like the point. We have all these big R01 university money or dollars, and we go and we give them a taste of some education that maybe they're not able to get as a supplement. The university itself is fantastic. There, it's just small. They just don't have the resources that we do. Um, Yeah. I get a light notification every time someone follows or subscribes. So I just got three in a row. No, no. And sorry, everyone. I didn't know it was so loud. Yeah, I see that. Yeah. And Gimpy G, who I recognize from your stream. So thank you. And then um, Manogard and Shazaberries. Okay, everyone's just deciding this chance to follow, which is fantastic. Um, yeah. And I'm getting double notifications on things. So mm. Yeah, that yeah, yeah. It's fascinating to me. I mean, because I only just started. It feels like I only just started my career in science. I mean, I went to college not knowing what PCR was, the thing that a lot of people now, due to COVID tests, know what that is. But absolutely, every other day in lab, it feels like I go onto my software and without thinking about it, I'm piecing together bits of DNA that I'm going to be ordering from it in a tube and shoving into an organism, and I've stopped thinking about it. Yeah, yeah, yeah. I mean, and speaking of just programming language, have you seen? I'm going to search DNA horse movie. Have you seen this? This was from several years ago. Yeah. Sorry. So a group, because it intuitively makes sense that you can do this, encoded into the DNA of an organism a video of a galloping horse. This one of these early movies. Yeah. I mean, that's not quite the programming language, but the ability, the fact that we have this capacity is crazy. Yeah. Yeah, no, so I don't know if you're viewing my stream, but I have a just a simple pop science article up about it. Yeah, no, this is completely designed. So they intentionally went in, they did this, threw it into some bacteria, waited several generations, and saw if they could decode what they had encoded as like a storage medium. Yeah, yeah, I think that's something that the book brought out. Yeah, I think everyone really wants to, but yeah, there's some. It's kind of weird because, of course, it's always George Church. Yeah, that's Watson. That's Watson. Yeah. um, Meanwhile, that's kind of what I'm going for. I can change my lights to be a little bit more dynamic. Honestly, I just like colored lights. That's all. There's no, yeah. There's a question from Shazaberries. Could you store a book you wrote in your own DNA like a biological copyright? So this is sort of an interesting question, but also an issue that at least I've seen with sort of thinking about storing information in DNA. Um, is you have to figure out how to what? I wouldn't be surprised. I mean, like you can do it. Absolutely, you can. But whether or not that gives you any sort of meaningful ownership over it might not, right? Like it's like we just invented the Gutenberg press. You know, if you put down your story in a book, does that mean you own it? It's like, well, it just takes a couple of years for everyone to be able to do that, so I don't really know. And the fidelity of getting it back, that's like or it would be your child would get some copyright. Yeah. Oh, so you're putting that into your germline. So that's bold too. Yeah. Yeah. Yeah, I'm finding that yes, a team encoded back in 2012 a 5.27 megabit book into DNA microchips. Uh, See, it's that we can do it, and then but microbloganism makes a good point that, and then hopefully your book doesn't happen to code anything in your DNA. So it's not a good idea to just throw in code because just by chance you're going to accidentally have a promoter and a start and a stop. You just got to hope it's not bad. Yeah. Yeah. Not on purpose, but by the emergent property of having a string of DNA in a cell, right? That exactly. We can. I mean, you can make random strings of DNA any length you want, and if it just so happens to look close enough like a gene to a cell, Now you've got some problems. Or worse, let's say you threw that on one of your chromosomes. First of all, bad idea, but let's say you just wanted to do it. Um, and you did such careful work to make sure there wasn't a single gene element in there, even by chance. But then you forgot that, well, DNA has intrinsic physical properties. And suddenly you've introduced something called like a fragile site into your chromosome. And a fragile site is exactly what it sounds like. It's a site that through certain cellular processes is prone to breaking. Well, now suddenly you've put in a weak point into your own chromosome, something that should not be there. And that could lead to a bad time too. I mean, there's so many different layers of just physically having a string of DNA where you shouldn't have it that would be a problem. I would think that generally encoding information into DNA wouldn't be something you'd want to put into your body. Yeah. I mean, we're awash with foreign DNA constantly. It's not like it's a huge issue to like spill some on your hands, but yeah. Yeah. Yeah, we really haven't specifically mentioned it. I've had it pulled up a couple times. And who was that you pointed at? um, okay, okay, I'll do it really quick, really quick. So this is the book that we've been offhandedly referencing, The Genesis Machine by Amy Webb and Andrew Hessel. Amy Webb, I think, is a futurist. Andrew Hessel is a PhD. He's a microbiologist and geneticist. And broadly, they talk about sort of the like recent history, because it wasn't that long ago, the present and the sort of future of biotechnology, specifically synthetic biology, all about genetics, more kind of hovering around that space. And then they also throw in some really interesting future semi-sci-fi perspectives of like, oh, this could happen. And then they have some recommendations at the end, some of which I agree with, some of which I maybe don't agree with so much. Um, but that's the long and short of it. A lot of just interesting information collected in this book. Anyone who is watching really should just go buy it and read it. It's a really good read, really well written. Hmm. Yeah, yeah. Actually, so someone, oh, thank you for the subscription. Um, so someone asked, yeah, it is. That's going into the new microscope pile. Yeah, fill it in a little higher. I do have to trudge kind of far to get down to it. And another follow. Uh, so there is a particular model. Like I said, trinocular. The model I have is some Chinese brand from the 80s. I can't find information on it. I just would like a more modern microscope with an LED light source, or at least the capacity for an LED light source. Right now, my halogen bulb, useful, really useful, but I tend to cook things alive because it gets so hot. Yeah, it gets a little hot when I rest my hands down. Um, well, well. I can pull out the electric contacts where the halogen bulb sits. I bought some LEDs, but they're the wrong shape, so I cracked off the outer layer, twisted the whole thing, and it sort of worked, just not as well as I wanted it to. Um, uh, yeah, someday though. The, Absolutely, but I know there's at least one person in the chat who will definitely tell me why halogen bulbs are still useful. It has to do with like the light range that comes off of it, because sometimes you want a more restricted or range of wavelengths that come off, or something like that. I'm not actually a microscopist by training at all. Okay, so I actually just wanted to quickly welcome Science Streams, but also go to something that my mod Defested said. The thought of having to say, please don't try this at home when discussing DNA manipulation is a wild thought and dangerously close to being needed. Now, I wanted to point that out because I'm just curious because this is brought up in the book, what our thoughts are on the idea of at home science and also the recommendation that they make, which is licensure for scientists or people doing research. Yeah. I'm not sure. I wouldn't be surprised. But the places that I go through, you do have to be from a university. Yeah. 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 DNA oligosynthesizers are just chemical machines. It's just fancy chemistry that's in a $10,000 box. So if you buy that box, you can do it. I, here. I'll close the camera. All right, just in case. Uh, is it with a Z? Josiah Zahner. Is that it? Yeah. Yeah, I mean the oh, yeah, it wasn't CRISPR based probably, or can't be. Yes. Yeah, I I believe at least one point they sold a CRISPR kit where it was like yeast, and what you were doing was repairing an already inserted but broken GFP, and it's effectively you were turning it back on. I don't know if that's still. I don't know if that was them or another company, but that's kind of the thing. Yeah, they use CRISPR as. I mean, it is CRISPR, but it's kind of a flashy title for something that. Yeah, yeah, absolutely. Yeah. It, yeah. 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 So someone just commented, Dr. Dahanoob, something that I think an opinion I share. I think the problem is he makes gene editing to be easy and safe and promotes that there is little risk involved. That is terrifying because there is risk. If you play with some bacteria, that's fine. But once he injected himself with some self-made CRISPR cocktail and told people that's fine to do at home. That's sort of like, yeah, yeah, yeah. No. And if he wasn't going to do that, be this person in our culture, someone else would have. So this platonic ideal of a DIY bio person, it was going to exist, I think. Right? It's we're too close to the head where this is common enough to do for that not to have occurred. Yeah. Yeah. Yeah, because if you have I mean, look at this. Well, maybe not if you're not like watching me right now, but everyone on stream, look at this. I mean, $40, and you get a couple petri dishes, some tubes. I think they have a kit somewhere. Oh yeah, right here for 200. You get a pipette, some gloves. You get the basics of a lab. Um, yeah, 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 yeah. It it feels strange to, because you want, like, I so much want to encourage curiosity in molecular biology. It's something you cannot see for the most part without really specialized equipment, but it's so cool what's going on just beyond our reach. But at the same time, yeah, I worry about the messaging. That's a good way to have put it. The like what you can do versus what you should do, not ought to do, but should do. Hmm. Yeah. Yeah. Eh, Yeah. It's interesting because in academia, I feel like you would be hard pressed to find too many scientists who would not freely and willingly share all the information they had to anyone who asked. I mean, there is a freewheeling spirit in the science too. Like there's a like GFP, right? Green fluorescent protein. It's technically patented, or at least a version of it is. Yeah, but a group of scientists made another GFP gene, right? They just modified it just so much, and they called it, because they hated that it was patented, and they called it FU GFP, free use GFP, and they give it out. They give it out to whoever will ask for it. But you're right. There are monetary incentives out there, especially when you get into industry, and that's how that's just how it works, unfortunately. No. Yeah. 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 Uh, that's literally a plot point in Foundation by Isaac Asimov. Yeah. So, but no, I mean, because there's truth to that, right? So, well, what does that? So then, do you think that James and I should have our be card carrying like licensed scientists? Yeah. I I don't know. Yeah. Hmm. 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 Yeah. Hmm. Yeah. 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 Which might have a place. But I totally agree with what you're saying, and I think there's a problem with sort of some of the language that people use, not to their detriment. It's that people talk to about biology like it's a plug and play sort of thing. Um, that's not necessarily true, especially when it's something new. So if you go into an engine and put a new gear in, you'll know exactly what that does. But when you're dealing with things in biology, you don't know what inputs and outputs that might have. And part of the education process as a scientist is partially to understand how much we don't understand. You can swear. You can swear. I don't care. Yeah. Ugh. Yeah. 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 One of my favorite cop out phrases for anything in biology is emergent property because it describes everything and nothing at the same time. We are conscious moving beings with self-awareness because some molecules hit each other the right way. Yeah. 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 Emergent property. Yeah. Yes. Uh, yeah. Oh, yeah. 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 Oh. Yep. Yep. Yeah. Hold. This is a nanopore flow cell. At one point, this could read megabases of DNA, just zipping it through a little enzyme. It's dead now and it lives on my desk. So Oxford Nanopore, I believe, uses some kind of like processivity enzyme to pull a string of DNA. I believe it. I believe they use changes in voltage. Um, in yes. So nope, it's in triplets. So that's why it's a little yeah, it's a little iffy in terms of the amount of fidelity that you get. In fact, that I've done nanopore sequencing before, and when you line up all the sequences, uh, I guess just to James and any other people who have seen sequencing, it looked like shit, the worst sequencing you've ever seen. But in aggregate, you can look down the line and say like, oh, that's an A. But it's awful. Besides that, yeah, it reads triplicate. Uh, it you have to do it in parallel because there's such a high rate of mistakes in reading. So you have to like, is that an A or is that a mistake? But if you do it 10,000 times, you can be like, oh, okay, that is an A. I know that for sure. So yeah, yeah, it comes up with some consensus sequence. Um, uh, you know, ooh, I don't have it on this computer, but I actually did have a slide showing some real results from one. So unfortunately, I don't have that on hand. But yeah, yeah, I'm trying to find just a good picture of this, to be honest. Oh, it's so small. It's so tiny. Yeah, so if people can see behind all these JPEG fragments or whatever they are, you just get a little protein and it sucks it through and it measures the current. And by reading that current, you know what base it is, more or less. Yeah, maybe. They're about $1,000 new. You can use them a couple times, but once it's used, it's just a piece of trash. Yeah. 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 He's yeah. No. Yeah. the progenitor sort of cases for these is the invention of IVF and also the invention of recombinant DNA. Both back, well, recombinant DNA I think was in the 60s or 70s. I don't know when IVF was, but it was within the same span of a couple decades. I mean, that was the start of it. That is, yes. So that is simply taking DNA from an organism, possibly mixing it up with another set of DNA from another organism, and maybe putting it into a like another organism. It's the idea that we can simply take the DNA away from where it was grown in nature and manipulate it to the point where there's still guidelines based on that sort of original set of recommendations, which the book does describe. But yeah, that was the start. That was, I think, I would guess if you went back to try and find the first instance of, scientists are playing God, it would probably be about then. Uh, yeah, the something sin 1.0, then they had 3.0. Yeah, that was many decades later. That was more recent. The Yeah, what is that called, James? Do you know? Cynthia. Uh, yeah, it is. It is. Yeah, yeah, right. Because they took an existing genome and basically popped out all the genes that they didn't think, yeah, were necessary. Yeah, yeah. Or it was supposed to. It was supposed to, right? Because yeah, and this is the weird thing about where some of this really interesting science is coming out of. It's like what they did was kind of simple scientifically. They took a whole genome, which we knew, saw all the genes, which we knew, and just deleted them one by one, and saw, okay, was that actually necessary? No, okay, chuck it in the in the basket, and then they just got rid of all the unnecessary genes or what seemed to be unnecessary. Like on paper, that's kind of an easy concept. It just took technology a little bit to catch up. And clearly, what they wanted to do was just like a couple years potentially down the line. I haven't heard anyone trying to do that, though. Yeah. Yeah. Hmm. Yeah. Well, you'll be happy. Yeah. Ah, nice. Yeah. Mm, I didn't realize you did that because meanwhile I am using the same protein. It's called TN5 transposase. I have a little 3D printout of one of a monomer of it. So I've tethered it to a nanobody, which is like antibody but smaller. It's from llamas, and I'm using it to go around and footprint in the genome. Wherever it sits down, it makes a little cut, leaves a little barcode, and I'll read that later after some sequencing. So 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 for James, obviously, it's about understanding sort of the like certain genes. What I'm doing is so that your DNA is decorated by hundreds of proteins, but we don't know necessarily where all of them are and when all of them are where they're supposed to be or when they're not. It gets very confusing, especially in metazoans, so animals, right? We develop, we change, all this tricky stuff that prokaryotes don't have to worry about. Um, by 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 looking around for where these proteins are, we can understand something about them. Trick is not quite because what I'm dealing with aren't ribosomal proteins, but it's the same concept. Yeah, it's a technique where it's basically like no microscope could ever see what we're trying to do. Rather, we're using this special protein as a tool to barcode the DNA where another protein is sitting down. And by doing that, we can infer something about its biology, where it is. Yeah. 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 Sure. Uh, TN5 transposase. Yeah. 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 Well, because the nanobody is specific to the protein that I'm interested in, so there's a certain amount of enrichment over the background. Yeah. So Yeah, go ahead. Yeah. A known, yes, exactly. Yeah. See, we're at the point where like this is just an idea I had one day, and although, like I said, we really shouldn't be putting together biology like they are little Lego bricks, sometimes we kind of can when we're using them as very specific tools. I mean, we can't use a microscope to look at these things, so we have to use what nature already invented via evolution as little tiny tagging markers or little things that chop things up. The resolution is not good enough, and we're talking genome wide in living cells. I mean, the whole nucleus would just, yeah, the whole nucleus would just, yeah, it's, yeah, yeah, yeah. No, so uh, nanobodies are single chain antibodies. It's an evolution of the camelids, so llamas, camels, alpacas. It is a part of their immune system. The key thing is that we can identify the known sequence of that protein, and because of that, we can produce them very easily. So no injecting like bunnies with your antigen or whatever. Yeah. So basically, the nanobody is to pair with a specific epitope, and since that's known, I can take my nanobody, uh, plop on some TN5 protein next to it and produce that protein myself in the lab. I have this little functional enzyme, and then I'm off to do my experiment. No, the nanobody does. So these are published nanobodies. That's why they're great because you can publish them as a sequence. Yeah, pretty much. Yeah. Yeah. I think see, I just use them. I don't know much about them. Yeah. Yeah, so here's a wonderful picture on my stream. Uh, all right. So conventional antibodies have this Y shape. There's this variable region up here where the cursor is. These little lumps indicate literally a shape, right? They fit into something. The problem with conventional antibodies is that there's two of them, and these are produced via this complicated mechanism. Whatever. Alpacas, llamas, and camels have single ones, right? And by using another technique to sequence the protein itself, we can figure out what that is, and because of that, we can chop off all the stuff we don't need and make a little functional unit. It is a perfectly shaped little thing that will find exactly what we need it to without too much extra faff attached to it. Yeah. A little detector. Yep. A little. I have my little detector and my little signal maker thing, my little barcoder. Yeah. Yeah. Gadgets. Yeah. 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 And this is the problem with, like, as you were describing before, this is kind of like the problem with education, right? Like so when I've done sort of work with children myself, I find that, like, kids, they're really able to understand molecular biology in a way that adults have a little bit of a harder time, because they just accept. They accept that that green thing right there has a shape and fits in with something else, just like one of their Legos. And that is how it works. It's just there's all this complicated stuff that adults try to put on it, and it makes it harder for them to understand. It's really a shame we don't get to it until late college, if you're lucky. Yeah. Yeah. No. Yeah, absolutely. Yeah. I, I mean, to bring it sort of back to what we were talking about a little while ago, with like the Odin company, the Odin, you know, CRISPR. I struggle with the idea that children should be thinking that they can do CRISPR at home in any capacity, but it's such a good technique for them to understand. Such a good, like, just like as a jumping off point into molecular biology. Like, you want to change something about the DNA. Everyone knows, kids know what DNA is because of Jurassic Park. And now we here's a tool that we can use to change it. They don't need to know the details, but it's so cool that that's, like, a kit, a thing. Yeah. Yeah. Yes. Hmm. Yeah. Yeah. A- And it's being warped, especially with the Uyghur situation in China. I mean, that's a sort of biological absolutism that also occurred, in the 1940s and late 30s, with Nazi Germany. I mean, yes, you can identify populations by SNPs. No, those do not mean anything meaningful for a whole species. Yeah. No. Yeah. 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 I wish that section was a little bit more expanded because I mean obviously they did their research. It's a little bit more well thought out and fleshed out than like the movie Gattaca, right? Because they clearly like what you just said, having this gene means that this other gene might not work so well. Like that's great. That's how biology works. But yeah, as we piece these things apart, it's just going to take a company, right? When's the first company that's going to advertise, Yeah, oh yeah. 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 Mm. Yeah. Because why not? Yeah. 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 Bye. Yeah. 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 And the the scenario you're also suggesting also is almost predicated on everyone having equal access to these tests and these abilities, which we do not, we never will probably, unfortunately, just because of the yeah. Yeah, exactly. Yeah. 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 And you're so right. There's like different. There's you've described perfectly the different levels, like within your family, within your community, your state, your country, and then finally like this. Not just the world, because that's a pretty static thing, but the species, plus and minus 10,000 years. But something that I won't argue for, but I think is important to remember is the idea, and we had a discussion on my stream a long time ago about this, the idea of transhumanism, the idea that even picking up a hammer, you have become more than human. We are a species dedicated to not staying in our lane, right? We invented fire. Now our guts are shorter than our hominid backgrounds. Glasses, yeah, exactly. So I don't want to argue that that means we are destined to now just use the newest tool. The newest fire is biotechnology, but that's, I think, a consideration nonetheless. We do this all the time. Yeah, yeah. I Yeah, mm, yeah. Absolutely. But there's one section that we didn't talk about, and I don't think we really have time to, but the section I want to mention that I'm most interested in, just for maybe actually solving more problems than it creates, is the idea of culturing animal products from a food perspective, because it gets into the problem of climate change, this whole other thing that normally biology can't, or biotechnology can't quite touch, or we haven't been able to. But ridding ourselves of factory farms and huge amounts of animal waste could be huge. Yeah, it's amazing. Yeah, yeah. You could meet you could meet the donor of the tissue in the restaurant, right? You could go pet the cow, say thank you for the biopsy, and then go. Yeah, yeah, Bioreactors. Yeah. Yeah. Uh, Meadery sounds great. Yeah. That was in this. Yeah. 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 You could you could do that. You could do that. Yeah. Celebrities sell access to their, yeah. 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 Hmm. Hmm. Really? Yeah. Interesting. Yeah. Oh, yeah. Yeah. That feels like a decade, maybe two away, you know. Yeah. Yeah. I feel like the graph that I always see about Canada is, oh, there's like most of the population is this one strip. In 50 years, I feel like it's going to be the other way around with the United States. Oh, most of the people in the United States live right up next to Canada. Ah, yeah, yeah. I do want to be cognizant of your time because it is three. Yeah. Okay. Do you mind if could you entertain one question that I promised I would ask from someone in the chat? This is from my mod Defested. He asks, David, which came first, your love of science or your science fiction career, and how did they affect each other? Oh, mm, yeah, yeah, yeah. Mm. Yeah. 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 Oh yeah. I think you're essentially like preaching to the choir, both here on stream and in the chat. I mean, yeah. Who? Oh yeah. Wait, me? Yeah. It's I made it on a so when I was a teenager, I watched Stargate. I got the box set of season 8 of SG1 for Christmas, very late. Well, we didn't have cable. So I got that for Christmas, and at that time I was just discovering the magic of coffee. So I made myself a coffee at like midnight. I dug out all the Legos, turned on the Stargate DVD, and in a fit of caffeinated productivity that I've never since had again, I watched the entire season, start to finish. I was surprised when I had gotten to the end because I was like, "Wow, that was short." Uh, yeah. It was the next day by the time I was in a basement too, so I didn't notice that the sun had come up and then been up for quite a long time. And in a fit of caffeine, I just made this little thing, and I don't know, I carry it around with me to places I live. Um, yeah, I'm just gonna set it to the side. It has a little stand. Whoops. Oh, yeah. Oh, those ones are so amazing. I'm a little afraid when you start saying Arduino or anything that makes me think that I have to put wires together. I get a little afraid. I should just go ahead and do it. No, that's just moving liquids into other liquids. I just press the buttons. Yeah, yeah. No, no, no. Yeah, I'm. Mm-hmm. Gel electrophoresis. Yeah, yeah, yeah. Yep. I'm one. Yep. I mean, I mean, no, no, no. It feels weird to say, but like you were one of many, you know, inspirations. It was more your way of displaying how to problem solve and that eureka moment, more than your treatment of other people that got me into science. Yeah. But absolutely, Mm. No. Yeah. Yeah. Well, it's fantastic. Yeah, that'd be amazing. I'd definitely like to. So, we should let you go. Um, you, yeah, yeah, yeah. So, thank you so much for joining. James, you too. Uh, okay, awesome. This was super cool, and we got through like 5% of the book. Not that we have to return to this, but there's so much stuff in here. Yeah, everyone should go read this. I mean, it's really good, or listen to it on audiobook. Yeah, yeah, yeah. Okay. Life. Huh. Actually, physics, quantum. Wow. Yeah. Yeah, that'd be great. Yeah, yeah, All right. Yeah. Great. Sounds good. Yeah. Thank you both for joining. Thank you for everyone in the chat. Don't all go away, but yeah, if you two need to leave, head out by all means. I'll close up shop. Just to close up. Just to close up. I swear. All right. Bye. Okay, sounds good. Okay, yeah. All right. All right. See you later. All right. Oh, you can still see me. Okay, cool. Wow, now I need to fix all this. This is going to go very badly very quickly because the call is going to drop. So let me just say, hopefully, what I need to say before it all automatically shuts down. Yeah. Thank you everyone for coming. That was awesome. You should definitely go follow. So James has a Twitch as well. Let's see. Shoot. James. There we go. Let me shout him out quick because I don't think. Oh, maybe not. If you do search James Gurney's username, you'll find him on Twitch, and then David Hewlett. The Twitch URLs were right there, so go follow them. I am just trying to get back to my dashboard so we can pick someone to go raid, because I think that's going to be it for me. I wasn't going to talk really anymore. Whoa, don't look at that. Look at the llamas instead. All right. Yeah, so if you're interested, if you're new here, so I normally, most often, are either on the microscope, playing around with some microorganisms, or talking slash discussing science and science fiction, usually around biology, the biology sort of realm of things. So this was very much on topic. So I'll be back on Wednesday. Let's find a channel to raid, though. Is Science on? Science was on before. I'm not seeing them. Okay, we're going to just raid Turtles and Chill. It's an aquarium. They're turtles. They're super easy. Stick around for that. Otherwise, I will see everyone on Wednesday. So thanks for stopping by. Bye.